Today on Film Science, we're going to transport proteins. So Science Understanding, we're going to look at, explain the roles of transport proteins, including channel proteins, such as aquaporins and carrier proteins. So what is a transport protein? Transport proteins are involved in transporting materials across the cell membrane. And we're just going to go through a few examples of transport proteins so you get an idea of how they work. Aquaporins are an example of a channel protein. A channel protein allows, with not much specificity, a substance through the membrane faster than it normally would travel through. And they work on passive transport. They don't require energy from the cell for the transport to occur. So aquaporins specifically allow for water to move in and out of cells faster than what would happen through diffusion alone. So diffusion is relatively slow because water is what we call a polar molecule. So chemistry students should know about this. It's a polar molecule, and the middle of the cell membrane is non-polar, so it's very hard for it to make its way through. It does, so osmosis can occur, but it's faster if you have a channel protein that has a width just about the right width for water molecules to pass through, and then they pass through the membrane a lot faster. So it allows for faster transport. Depending on the aquaporin that we're looking at, some specifically just allow water through, some can allow some ions to come through as well. But when we're thinking of aquaporins, we're thinking of water channels. We're going to have a look at these a bit later on when we look at the kidneys. Carrier proteins are a little bit different. They can be involved in either facilitated diffusion or active transport. What they can transport is very specific. So a specific protein would be able to transport one type of sugar or one amino acid. One nucleoside. A nucleoside is a component of a nucleotide. Now, whether the carrier protein is involved in active or passive transport, it depends on the material being transported into or out of the cell. As I said, they're very selective, so a specific protein will be able to transport a specific substance, or maybe a couple of substances, depending on which protein we're looking at. So here's a way of comparing them. So here we have our channel protein, so those substances are passing through readily, and it's not very specific. Whereas with a carrier protein, they're very specific, so here we're only picking up our little hexagons here and transporting them in. In this case, it looks through its facilitated diffusion because it doesn't look like energy is being used in this case. So let's look at some examples of carrier proteins. So the glucose transporter is a good example. So we're looking at GLUT1 here. So this is GLUT1. So this carrier protein specifically transports glucose with the concentration gradient into cells. I think it's specifically into rather than out of. So we have a glucose in the outer extracellular space. It gets transported through the protein inside the cell. So it binds to a site inside the protein. When it does that, the protein changes shape, and that allows the glucose to come off of the protein, and then now it's inside the cell, so it's in the intracellular space now. When it disconnects, the protein snaps back to the shape that it was initially, and now the next glucose molecule can come through. This doesn't require energy from the cell. It's facilitated diffusion, and diffusion is a passive transport process, but it's facilitated because we're using a protein to do this job. Sodium-potassium pump, on the other hand, this uses active transport. So in active transport, we're moving substances across the membrane, and we're using energy to do that. In the case of the sodium-potassium pump, we're using ATP. Ions are usually somewhat tricky. Again, they have a charge on them, and the middle of the membrane, so here's our cell membrane, the middle of it is non-polar, so it's tricky for polar things or really highly charged things to pass through easily. So the job of a sodium-potassium pump, we're moving the ions against their concentration gradient across the cell membrane. As I said, because it's a pump, we're pushing things against the concentration gradient where they normally wouldn't go, so we have to use energy. In this case, the energy is in the form of ATP. What happens is some sodium ions from inside the cell attach to the sodium-potassium pump. An ATP molecule also attaches to the pump at the same time. When the third phosphate group breaks off, some energy is released that can then be used to change the shape of the pump slightly. When that changes the shape of the pump, the sodium ions are kicked out of the cell. They go to the extracellular space. The changed shape of the protein allows for some potassium ions, that's the K pluses here, to attach to that pump. And when they attach, the protein changes shape, and that allows the potassium ions to come into the cell. The phosphate group then also breaks off, and then that can be regenerated into ATP using energy from the cell on aerobic respiration. So what happens is we're pushing sodium ions out, and we're pulling in potassium ions, and that sets up a concentration gradient inside the cell. These pumps are crucial in nerve cells. Nerve cells use electrochemical pulses to transmit information across the body. And part of that is a change in polarization of the membrane of the cell. And these sodium-potassium pumps are really important in that. The last one we're going to look at is the glucose-sodium transport protein. Again, this is an active transport process, but it doesn't use ATP. It uses a slightly different way of moving substances against the concentration gradient. What this means is if there's already a high amount of glucose inside a cell, we can actively pump more glucose into that cell. So it goes against the concentration gradient. So we get higher concentrations of glucose inside the cell, and if this is happening in the kidneys, which is a common place for this to happen, the glucose can then pass through into the extracellular space and then pass back into the blood, so that we don't lose glucose that would normally get filtered out of the blood at the kidneys. It doesn't use ATP, this process. What happens is glucose and sodium's attached to the inside of the protein, and then it changes shape again slightly and the glucose comes in. The sodium ions set up an extra positive charge inside the cell, and that extra positive charge is enough to generate the movement of the glucose through. So it's a bit more of a complicated process, not just using ATP, but we're setting up a charge differential which leads to the active transport of the glucose into the cell where it normally wouldn't happen. So let's do our check. You need to explain the role of transport proteins, including channel proteins such as aquaporins and carrier proteins. So today on Flipping Science, we looked at membrane proteins. That's it for this last day. See ya.